Good morning. Today my class is on the topic of exome sequencing versus targeted gene analysis in liver disease. So the objectives of this class will be first to have a basic overview of the human genome, then look at the methods for targeted genetic testing, look at next generation sequencing, and then we'll look at literature comparing targeted versus next generation sequencing in relation to liver disease. So the human genome, here we'll concentrate on the nuclear genome. It is packaged into 46 chromosomes, which consist of 22 pairs of autosomes and 2 sex chromosomes. So it has around 3 billion base pairs, and they code for around 20,000 genes. These genes this account for only around 1 to 2% of the whole genome, with the rest being introns, regulatory sequences, non-coding RNAs, and repetitive regions. So as we see, though the entire genome is large, around over 3 billion base pairs long, it is divided into functional units called genes, and these genes are made up of introns and exons. So the exons are the functional unit which is then translated into proteins, and these consist only about 1 to 2% of the genome. However, they account for over 85% of all recognized disease-causing mutations. So hence it becomes both cost-effective and also easy to analyze just the exome to identify most of the causative mutations. So the first most common technique used in genetic laboratories for genetic testing is the polymerase chain reaction. So this is a technique in which we use to isolate a particular sequence of DNA, form a complex DNA mixture of DNA. So this requires primers which are designed to bind to the DNA complementary to the target region we are interested in, deoxynucleotide triphosphates and a polymerase. So these primers go and bind to the target region, and polymerase along with the DNTPs form complementary strands. So this cycle goes on, and we keep getting new copies of the DNA. And so normally this process is run for around 20 to 30 cycles to get a large quantity of the target enriched region. So there are multiple variations of the technique, and a variation of this technique called ARMS PCR is used for the targeted testing of Wilson's disease for these two mutations, which we are currently doing in the institute. Another technique which is commonly used is restriction fragment length polymorphisms. So this utilizes the principle that there are a number of endonucleases which are produced by bacteria, and these go and cut the DNA at specific restriction enzyme sites. So these sites are well known throughout the genome. So we utilize this knowledge of the location of these sites in order to design the tests. So if we can design a test with a mutation affecting one of the sites, we can end up getting a different lengths of the fragments, and this difference in the length is used to detect the disease. So this technique is used for these 3 common mutations for Wilson's disease tested at the institute. The next technique is Sanger sequencing. So this is the gold standard technique for DNA sequencing, developed in 1977 by Frederick Sanger. So it is based on the principle of selective incorporation of dideoxynucleotides. So these differ from the normal deoxynucleotides in the fact that once they get incorporated into the chain, they do not allow any further incorporation. So these dideoxynucleotides are mixed in with the normal deoxynucleotides into the mixture, in the reaction mixture, and they compete with the deoxynucleotides to get incorporated in the chain. So at the end of the cycle, we end up with chains of different lengths, each terminating with a dideoxynucleotide, and each of these dideoxynucleotides are labeled with a specific dye, which then can be used either in a capillary electrophoresis in a sequencing machine or on a gel electrophoresis to identify the particular terminating nucleotide. This is then put together in order to identify the entire sequence of the target region. So normally this reaction can only do one sequence per read, and each read can be between 500 to 1000 base pairs. So the indications of Sanger sequencing would be in disorders in which there's a clear phenotype, that is the clinical features point to one known disorder in which there is a single gene, and this gene should be either small or it should have a hotspot region, or we can use it to confirm our next generation sequencing reports, or if the mutation is already known in a family, then we can just do this targeted Sanger sequencing. An example of a disorder which is commonly tested by Sanger sequencing is beta thalassemia, since it has only 2 exons, so it's easy to do this technique. Next, coming to next generation sequencing, this is a technique which was developed in the 2000s. So this has really revolutionized the field of genetics. So it uses a principle of massively parallel sequencing, in which millions of DNA particles are sequenced at the same time, and so this has both high throughput, both in the amount of DNA sequences which can be sequenced together, and the number of samples which can be sequenced at the same time. This has dramatically reduced the costs of sequencing, which has dropped from over 100 million in 2000 to around $1,000 for a genome at present. So the basic steps of exome sequencing is we get the patient's DNA, which contains both the introns and exons and non-coding regions. So in case we want to do a custom panel or a exome sequencing, then we'll only want to capture the regions of interest. So we'll use a exon capture kit to capture the regions of interest. And once we have the regions of interest, these DNA fragments are then processed, means adapters are added, and then these are attached to specific flow cells where they undergo a PCR in order to form particular clusters. Each cluster corresponds to one original DNA fragment. So a flow cell will contain millions, tens to hundreds of millions of these clusters. Then the actual sequencing process occurs using deoxyribonucleotides with specific dyes, and the machine captures an image at each step in order to build up the sequence. So that was the the steps which occur in the lab. Next process would be the bioinformatic pipeline. So this generates a large amount of data, and this data has to then be processed through bioinformatic pipelines in order to get useful information. So there are a number of steps, and in the end, the process is that we take each read and try to align it to a particular reference genome, and then so each of these gray color bars are one read from the original data, and so these are aligned to the reference genome. Here's the reference genome, and then software is used to detect any changes from the. So here we see there's a heterozygous change which has been picked up by the software. So normally when we do a whole genome analysis for everyone, we'll get around 4 million variants. If you do a exome analysis, you'll get around between 50 to 80,000 variants, depending on how you've designed your exon capture. And so this is a large number of variants, and now we have to filter them down to find a one or two causative variants. So we run a number of steps in which we first filter out the common variants based on public databases, then depending on the function of that region, and depending on the inheritance pattern, and then we try to cut it down to one or two which will explain the phenotype. So the scope of tests which can be done through next generation sequencing could be targeted panels, and these may range from a few genes, for example, like in the PFIC panel, or several tens to 100 genes like in a neonatal cholestasis panel, or several hundred genes like in an inborn errors or metabolism panel. A clinical exome is a common test offered by a number of genetic laboratories in India, which is commonly used. This consists of testing about 5,000 to 6,000 clinically relevant genes. These are genes which are known to cause diseases. The whole exome is testing all the exons of all around 20,000 genes, and then there's a whole genome sequencing in which you test both the exons and the introns. So a brief comparison between this. As we said, the panels only cover a small number of genes, whereas the whole genome covers both the coding and the non-coding regions. Limitations of panel testing in clinical exome is that it's only limited to the number of genes which are covered in the panel, and it will not detect variants in any other genes. Whereas a whole exome gives you the possibility to detect variants in genes which have not yet been implicated in any clinical disease. Whereas whole genome will allow you to detect variants both in the intronic and also exonic regions. However, data interpretation and interpretation of variants in the intronic regions is difficult, and it's currently very costly. Also, it generates a large amount of data, like each genome will generate about 90 GB of data, so it's difficult to both process and store this information. Currently, whole genome sequencing is currently used mostly in a research setting, whereas the other three are currently used in a clinical setting. Additionally, whole genome sequencing can detect chromosomal abnormalities which are not normally detected in the other techniques. So the indications of exome or genome sequencing, according to American College of Medical Genetics, would be to do the testing if there's a clinical diagnosis which is suggestive of a genetic etiology, but a targeted testing is not available, or the patient's disorder is highly genetically heterogeneous. That is, that there are a number of genes that might be responsible for the clinical phenotype, or if targeted testing has not resulted in any diagnosis. An additional point would be to do NGS in situations where the cost of doing the NGS would be less than sequencing the individual candidate genes. And now, because of reduction in costs of this technology, often it's cheaper to do the NGS rather than do individual gene sequencing. So now we look at the question of whether we should do with targeted testing or whole exome sequencing. So I'd like to use the example of, so it's difficult to generalize this because each disorder is different, and but I'd like to use the example of Wilson's disease. So Wilson's disease, we see there are it has been an example of using targeted testing because there are certain mutations which are present in high frequency in the population, and so targeted testing could pick up a large component of those mutations. So if you look at this map, we see that in a lot of the European countries, the common mutations account for nearly over half, or around half to over half of the all the disease variants. So this makes it a good disease to do targeted testing. However, if we look at India, the situation is not so clear-cut. In India, we see that the common mutations don't account for such a high percentage. They vary between 10 to 15%, and these also there's variation between the different geographical regions in India. So this makes doing targeted testing might not be as useful in our situation because we'll only pick up a smaller percentage of the population using this technique. Also, the gene is a large gene containing 21 exons. It has over 650 likely pathogenic mutations, plus an additional 600 variants of unknown significance, which have already been reported in literature. And most of these are seen in single families. So doing a technique like Sanger sequencing is very cumbersome and time consuming. So using next generation sequencing could be useful in detecting the variants. So there's some literature on next generation sequencing in Wilson's disease. In this study from Poland, in which they did testing on 248 patients, they detected mutations in a large percentage of the population, and only 3 patients did not have any identifiable pathogenic variant. This showed that this was a good technique to identify the mutations. Additionally, they were because they had done a whole exome, they had information on other genes also available, so they could look at possible genetic modifiers, because as we know, the phenotype is quite variable, with some having neurological and hepatic disease, and also variability with the onset of disease. So they were able to identify certain genes which they thought could be modifying the phenotype of Wilson's disease. So if we look at the use of next generation sequencing in pediatric liver disease, first we'll look at gene panels. There's been a lot of literature looking at gene panels in childhood cholestasis and also inherited pediatric liver disease. So so we see there are number of studies from different countries around the world, and they have reported diagnostic rates ranging from 17% to 68%. One factor which you can see that as the size of the gene panel tends to increase, the diagnostic rate tends to also increase. So because as we include more genes, we are increasing our sensitivity of the test. So there are some recent studies looking at the combination where they have used both gene panels along with whole exome sequencing in pediatric disease. So this is a study from China in which they divided the groups. So in patients which had a strong suspicion of a genetic disease that it fitted into a particular phenotype, they did targeted testing. However, in patients in which there was non-specific features, they did a whole exome sequencing, and overall they had a diagnostic yield of 55.8%, and their diagnostic yield in whole exome sequencing was 47.2%, and targeted testing was 62. And this was not statistically different. Though this difference could be explained by the fact that since they had done the targeted testing in patients which had particular high clinical suspicion of a particular disease, definitely in those situations your diagnostic rate will also be higher. Similarly, in Italy, there was a study in which they followed a diagnostic algorithm for neonatal and infantile cholestasis. So in this, they enrolled 50 patients. 38 patients underwent panel testing. In this, they did a virtual panel. So they did a whole exome sequencing, but then they only analyzed a particular set of genes, which they called a virtual panel. So 38 patients underwent panel testing, 6 underwent trio whole exome, that is whole exome along with their parents, whole exome, and 6 did single gene confirmatory testing. So overall they had a similar diagnostic rate of 60%. For the whole exome, they identified a mutation in 5 of the 6 who underwent, and in the panel testing around 50%, slightly less than 50% of the patients were detected to have mutations. Another study I would like to highlight was this study from Pakistan, in which they predominantly did exome and genome sequencing in individuals with genetic disorders. Most of these patients were patients with hepatic phenotypes, since most of the samples were sent from a pediatric gastroenterology unit. And overall diagnostic yield similarly was also 61%. One important feature of doing whole exome and whole genome is that in 12 patients they were able to identify mutations in candidate genes. So these are genes which have not yet been reported as part of reported as implicated in a particular phenotype. One important gene was this USP53. At a time when they detected it, it had not yet been implicated in cholestasis. So this would be an important use of exome sequencing or genome, was that to discover new gene disease associations. Now looking at adults, we see that the literature is, there's less literature of the use of next generation sequencing in adult populations. However, recently there are a number of studies, so I just highlighted a couple out here. So in this first study, in which they used whole exome sequencing along with deep phenotyping for 19 untreated adult patients with idiopathic liver disease, they found 4 monogenic disorders in 5 of the patients. Two of them had MDR3 related deficiency due to mutations in ABCB4 gene. One patient with a which was clinically classified as non-alcoholic steatohepatitis was found to have a mitochondrial disorder due to homozygous pathogenic mutations in NDUFB3. Along with one patient each had a disorder in type 3 familial partial lipodystrophy and hypercholesterolemia. And all important pointer was in the study was that all of these mutations, all of these genes identified were actionable. So they all of them underwent a change in the management post the genetic testing. Another study which looked at NGS, you did a targeted panel for the PFIC genes in cryptogenic cholestasis in young and adults. And in this study, they found pathogenic or likely pathogenic mutations in around 20% of their population. And there's a study looking at use of NGS in patients with NAFLD and hepatic carcinoma. So they had a cohort in which they had 142 patients with NAFLD and hepatic cell carcinoma, 59 with NAFLD and advanced fibrosis, 50 controls, and additionally they looked at data from 400 healthy controls from the 1000 Genome Project, which is an open source resource. So they identified genetic Mendelian disease which predisposed to either a cancer or a liver disease in 11% of the total subjects. Additionally, they did some statistical tests too, and they found that there was significant enrichment of variants in genes which affect telomerase, the telomerase or DNA oxidation, and involved in cell growth and proliferation, or in hepatic lipid metabolism, which could be related to the cancer. They additionally tried developing a genetic risk score using this exome sequencing data, and they found this score to be better for performing as compared to only doing the common PNPL3A mutation or the PNPN3A and TM6SF2 mutation, which are common, which have been previously implicated in hepatic cell carcinoma in patients with NAFLD. So here you see the area under curve is higher in this. However, although they found a statistically significant difference when using both the clinical plus the genetic risk curve compared to clinical alone, this difference is not that significant. So points to remember while sending genetic testing. One important thing is aspect is this first important thing is that is this the right test for the disorder. So if you are doing a next generation sequencing, it has limited ability to pick up chromosomal abnormalities such as deletions or duplications, and it won't detect deep intronic or intergenic variants. So in case you're doing you're doing a testing for mutations in these regions, then this test might not be the ideal test for you. Then another important aspect is if we are doing panel testing, we should always confirm if the gene required is included in a panel and what is the coverage of that gene in the panel. So there are certain genes which might not be covered completely 100% because there are certain regions of DNA which are not properly sequenced. So it's important to check that before we send a test. Important for counseling the patient is knowing the yield of the test. So because since these tests are costly, it's important to explain to the patient that okay, so and so test has only a 40% or 50% chance of identifying a mutation. Should know the cost and the turnaround. The turnaround is important in disorders in which we need to take some clinical decision. This is particularly important in cases in the neonatal ICU or pediatric ICU where quick interpretation and reports is important to change the management. And finally, these tests require proper interpretation because variants of unknown significance or genes of unknown significance are difficult to interpret and hence counsel the patients. They might identify incidental findings, that is mutations in genes which are not responsible to the phenotype, and also there's important concerns regarding data storage and privacy concerns. But in the end, the most important point is that there should be good phenotyping, that your clinical examination should be very good prior to sending the genetic testing, because especially for next generation sequencing, if you've given a wrong clinical data, your causative variant might get filtered out or it might not be looked at. So a testing strategy could be a patient who has come to the OPD and has a after clinical history and examination, you have a suspicion of a genetic disorder. And after ruling out secondary causes, if there's a clear phenotype or a strong clinical suspicion of a particular disease, for example, like Wilson's disease, then you could do targeted testing or an NGS panel or exome, if based on cost and availability, because not all genes might be covered in a targeted testing. However, if the phenotype is non-specific or it has multi-system involvement, then it would be probably better to go for a whole exome sequencing. And after doing this, if we still don't find variants or find only a single variant, then we would need to do other tests for chromosomal abnormalities, which are not covered in this lecture. And if in the history and examination we identify there is a clear-cut genetic diagnosis already known in the family, then we don't need to repeat the exome sequencing. We can straight away go and do a targeted testing for that particular variant. So in conclusion, NGS has revolutionized the field of diagnostic and genetics. However, interpretation of results needs to be done very well. The clinical phenotype and clinical examination is very important to identify the proper diagnostic variant. So it's important to remember these laboratory techniques are only an adjunct to the clinical diagnosis, and you should take efforts to match the genotype and the phenotype to make sure they are in sync with each other. Thank you.